Okay, so for the next section in the chapter, we're going to be discussing in section 6.3, we're going to be discussing bacterial growth. Now remember, when we're talking growth here, we're not talking increasing in size like you grew. We're talking about, with microbes, we're referring to growth. We're inferring an increase in population. Now, with bacteria, growth is by what is referred to as binary fission. Binary, two, fission, splitting. One becomes two. Thing to keep in mind, each of those resulting two cells, and over the next generation and generation, they are all clones, genetic copies of the first cell. You'll see in the textbook and other textbooks, and you'll hear me refer to it, the nomenclature is the parent, the one cell that starts that new culture, starts that growth, and the resulting cells are the daughter cell. So the one cell that splits gives you two daughters. Half an hour later, those two daughters, the one that splits, the parent giving the two daughters, and so on and so forth. Now, this isn't mitosis like you've learned with eukaryotic cells. What you have here is a complete division of the cell, and the only thing the bacterial cell has to worry about is the duplication of the one circular genome. Everything else gets divided between the two resulting cells. Now, as you see here, this one original parent cell will increase in size somewhat. It will duplicate the chromosome. It will make extra ribosomes, and then it's just going to start building a wall between and down the middle. As that wall continues, the cytoplasmic membrane will get pulled in, it will get pinched. And as it gets pinched further and further and further, it gets stretched until it's about to tear, and then it just resolves into two new membranes. One cell becomes two.